Good evening, everyone, and welcome to this evening, which I think will be very informative and educative for all of us. The title of this evening is COVID Immunity and Vaccination, particularly looking at COVID vaccines and insights into our immune system and how vaccines work in our bodies. And before I start, I first of all want to thank Julie Conn, Chair of our Education Committee, who absolutely led on this project, as she does on so much else. So much of the incredible educational activity that takes place in our community is under her leadership and through her initiative, and this evening is absolutely part of that. So even though I'm fronting this, I really want to just start off by acknowledging so much of what she does within our community. And I really want to welcome all of you to this evening, and of course in particular to welcome and thank our presenters of this evening, who I will introduce in a few moments. The format of the evening, though, will be that we will hear from both Professor Benny Chain and from Professor David Katz. We'll start off with a recording that Professor Chain has put together, in which he'll speak about the science of vaccination, following which each of our honoured guests will speak a little bit about their area of experience, and there hopefully will be time for a question and answer afterwards. I followed the normal format of muting everyone. However, at the end of the session during the Q&A, please do feel free to unmute yourselves, or equally to ask questions via the chat, which you can post either to everyone or post to me personally, and I will pass on your questions and moderate the Q&A in order that it can work efficiently. So that's the format for this evening, which I'm really looking forward to learning about an area that we're all perhaps hearing about a lot in the media, but sometimes struggle a little to understand. And so without further ado, I will introduce our guests. First of all, Professor Benny Chain. On a personal note, actually, as you will remember, Professor Chain, earlier on in my rabbinic career, I had the great pleasure and privilege of being acting rabbi in Dunstan Road when Rabbi Belowski was on sabbatical, and you were the chair of the shul at the time. So thank you for introducing me to shul life and working very closely with a fantastic lay leader. Professor Chain obtained his PhD in comparative electrophysiology at the University of Cambridge. And perhaps actually, before I go further in speaking about the biographies of either of our guests, I should say there are a number of words in their biographies that I'm not sure how to pronounce. So please forgive me in advance. Professor Chain also studied comparative invertebrate immunology in New York, and then worked in the UCL, becoming Professor of Immunology in 1996. He also moved over into, worked as a visiting professorship in the Weizmann Institute in Israel, working with systems immunology there. And in the last decade has realigned his research to exploit mathematical and computational approaches to make sense of the data explosion generated by the functional genomics revolution. And his research has focused on using high throughput sequencing to analyze and model the T-cell receptor repertoire in order to understand and manipulate the immune responses to HIV, to cancer, and various other phenomena. So I believe, Professor Chain, it's accurate to say that your area of expertise at the moment is computational immunology, not an area that I knew existed until I read up a little bit about some of the research you are doing. It is also really a great delight and honor to introduce Professor David Katz. Before I speak about David Katz's professional experience, I think really just on behalf of the whole of Anglo Jury, we owe David Katz thanks. He is really an unsung hero of Anglo Jury, who works with great modesty behind the scenes in an enormous range of areas that are of absolute vital necessity to our community, and uses his expertise in medicine and in the sciences in general to represent our community, to work at the highest level with government, and to ensure that a lot of areas that are really vital to Jewish life are able to continue. So even before we speak about your professional expertise, thank you so much on behalf of all of us, Professor Katz, for that work that you put in. Professor Katz studied medicine in the University of Cape Town, working in pathology and hematology in Israel, and then completing his training at Barts. He's been at University College London and related hospitals really ever since then, I think now for some 4 decades, as a research fellow in zoology and diagnostic service, and worked actually together with Professor Chain in the area of antigens and how they're introduced into the immune system. I think a particular area of interest is in cells which specialize in this role, dendritic cells, which perhaps we'll hear more about later, and how the immune system can go wrong. So again, we are very much interested in hearing about your area of immunopathology and your expertise in that. So thank you both of you so much, distinguished guests, for joining us this evening. And I'm now going to hand over to a virtual Professor Chain, who through the recording will speak to us for some minutes about the science of vaccination. I'm going to be sharing the video. Please excuse me if this doesn't work well instantly. This is a little bit of a new experience for me. I will share the video screen and hopefully you'll also be able to hear sounds. If you can't hear sound after about 15 seconds, please just let me know via chat. Okay, hopefully this will now work.
Hello, I'm Benny Chain. I'm Professor of Immunology at UCL, and I wanted to take a few minutes of your time to explain what vaccines are and how they work. We are all very aware that as our children grow up, they face all sorts of physical dangers in their environment, which they learn by trial and error, partly to avoid or to deal with. But of course, we also teach them, we educate them to recognize danger and stay away from it. It took the human race much longer to realize that we are also faced with a different sort of danger, an invisible danger in the form of billions of microorganisms which live in our environment and on occasion can enter our body and cause disease. Remarkably, our body has a system, the immune system, which can learn to recognize these microorganisms, distinguish those that really pose a danger to health, and eliminate them or prevent disease. The system is very efficient, but it's not 100% efficient, and many children still die of infectious disease before their immune system has learned to work effectively. About 250 years ago, we learnt that just as we can train children to avoid physical danger, so we can train our bodies to recognize dangerous microorganisms and respond to them and prevent disease. This process is called vaccination. And today I'm going to explain to you how the immune system actually recognizes these invisible dangers in our environment and how we can improve on natural immunity by a process of vaccination. So I'd like to show you a little bit about how this system actually works. And I'm going to use for it a diagram which was drawn just over a century ago, around the time of the First World War, by a German Jewish scientist called Paul Ehrlich, working in Germany. And he wrote a book about what was then called the immune system. And I'm taking a picture from that book, which I'm going to show you, because the principles that he put forward then, long before he really understood how they worked, turned out to be extraordinarily prophetic. So if you look at the left-hand picture here, you'll see these round things. These are how Ehrlich imagined our white blood cells to look at. We call them now lymphocytes, but they're basically cells in our body. And remember, these pictures, everything I'm talking about, it is at an immensely microscopic scale. So the real size of everything on this picture has been magnified by well over 1 million times in order for us to see it, and I can show you on the picture. It's completely invisible to the naked eye. So here are white blood cells, and he was particularly concerned at the time with diphtheria, which was causing a huge amount of disease at the time, and he pictures the diphtheria. Diphtheria was known to produce a poison which killed people, basically, if you got diphtheria. And he shows this as this black thing. It's kind of the evil toxin up here, and you can see it here. And he imagined that there are also other bacteria around. This might be tetanus, for example, trying to kill you. And each one, he imagined, had slightly different shape. These objects we now know are called proteins. They're tiny, tiny objects, millions of times smaller than we can see, and they cause the damage by latching on to parts of our body and damaging them. What he put forward, and this was his great insight, was he believed that these white blood cells had on their surface other objects which were varied in shape, but of which there would be at least one which fitted, almost like a hand in a glove, an exact match for the shape of this particular object which was attacking the body. Could be a virus, it could be a bacteria, it could be a poison, it could be anything. So there's a kind of a key and lock, if you prefer that analogy, between these two things. He called these objects, which were being produced by the cell, antibodies, anti-against, because they fitted against this object that was attacking them. And what he suggested was that the white blood cells would patrol the body. When one of these dangerous objects was exposed, entered the body, for example, you scratch your finger on a nail and the bacteria enters into the blood, then there would be an exact match between this object, the bacteria or the virus, and this antibody, this object on the white blood cell. And you can see how they match exactly. Of course, there would be this is a highly specific interaction. So this would work fine for this particular bacteria, wouldn't work so well for this one, which he pictures as being a different shape. And so there would be a different object for this. The trick is the following. When this object interacts with the white blood cell via this interaction shown here, the two interact like a hand in a glove or a key in a lock, then the cell, the white blood cell, produces millions of these identical objects and floods them into the blood. And it produces so many of them that they are able to mop up this dangerous molecule or this dangerous virus by basically sticking all over it and what he called neutralizing. In other words, the dangerous object, whether it be a bacteria or a poison or a virus, could not carry out its task. It could not cause any damage because it was completely covered, plastered with these antibodies, objects produced by the body, by the white blood cells, which block them. And so you would block infection. And what's more, these objects, he surmised, and indeed it turned out to be true, would hang around for weeks, months, or years in the blood, so that if you were exposed again to the same object, then the body would be pre-warned and immediately be able to block the invasion of this organism. Now, under normal circumstances, what would happen would be there'd be a kind of a race. The bacteria would try and grow and cause damage as quickly as possible. The body would try and fight against it. Usually the body wins, but as I said, in many cases, there wasn't enough time to make these objects. And so the bacteria wins and the body succumbs, or the human person who was infected would succumb. The trick of vaccination is very, very simple in essence. By exposing the body to something that looks very similar to the original object causing the damage. So in the case of, let's take another example. Let's take polio, which was a devastating childhood illness until maybe 50 years ago. That polio, you cannot expose, obviously, the body to polio itself. But the trick of vaccination, the insight was that if you took just one part of polio, perhaps its outside surface, so it couldn't cause any of the damage that the virus itself did, because it's really only a little part of it, and expose the body to it, it would be tricked into believing that it had been exposed to polio, and it would produce these antibodies which would then block a future infection with the real polio itself. That is the principle of vaccination. And essentially, that's how vaccines work in every case that we know of today. And that is the principle on which the current attempts to build a vaccine against coronavirus. Again, we don't want to expose the body to coronavirus itself because that causes damage before many people can protect themselves. Of course, in most people, the immune system works fine and they get better, but in some people it doesn't. But we expose the body to just one part of that coronavirus, and it's sufficient to trigger the body's memory. And note that this vaccine is highly specific. By producing these antibodies, you will be specifically protected against these sort of organisms shown here in black, but you will not be protected against this white one, for example, that has a different shape. So by getting a vaccine against diphtheria, you cannot be protected against polio. By getting a vaccine against polio, you won't be protected to diphtheria. Each one of these vaccines is entirely specific to the organism, or at least a family of organisms which caused it. That's really the principle of vaccination in just a very few words. I just want to, we've been sort of inundated with statistics and numbers, I think almost saturated with them. I just want to finish with one such number. Smallpox, perhaps some of you will still be old enough to remember the days before smallpox vaccination. But I wonder how many people are aware of the scale of the problem. In the years between 1900 and 1967, we estimate that 300 million people, most of them children, died of smallpox worldwide. 300 million people. And it's a horrible, horrible disease. You see here a picture of a child infected with smallpox. Now, we had a vaccine already, but it was not, it had not been rolled out on a worldwide scale. In the 1970s, the World Health Organization rolled out a world immunization protocol against smallpox. And as a result, by the late 80s, smallpox had been completely eradicated. In the last 20 years, there have been 0 cases of smallpox in the world, an astonishing feat. So just let me finish with the idea once more that I started at the very beginning. Just as we can educate our children to avoid the obvious dangers of growing up, so we can educate our bodies in exactly the same way to warn them of dangers at the microorganism level and to protect them from these infections. Thank you.
Thank you very much for that presentation. And now, Professor Chain, I'm going to hand over to the real you.
Thank you very much. So just a very few minutes, because I want to leave time for questions and answers at the end, and for Professor Katz to also to speak. But just as the Midrash tells us that when Yaakov went to meet Esav in battle, he had kind of 3 strategies that he would adopt in case one didn't work. So the immune system is more than just antibody. And I just want to very, very briefly show you the other arms of the immune system. There are 3 actually major arms. So if I just share my screen. Can you all see that? I hope so.
Yeah.
So let me just. Yeah, so the first one, the one that's been most studied, since, as I say, Paul Ehrlich started it, really the proper study of antibody was antibody. And I just wanted to start by showing you this picture. This is a diagram of SARS-CoV-2, the current, or COVID-19, as it's sometimes called, the current SARS virus. And you can see it's a kind of this is a cross section, so you can imagine it as a tiny, tiny sphere. It's about a millionth of a millimeter or something like that in size. And it's on its surface, which actually gives it a name, you can see these objects, and these are the proteins I was telling you about sticking out. They look like this. They're sort of upside down kind of cone things. This is the virus, and this is them sticking out. And this is a picture taken at a tiny scale, where the gray is this surface of the coronavirus, and the green are these antibodies, which Paul Ehrlich was speaking about so many years ago, stuck to the surface. And you can see that by sticking to the surface, they prevent the virus interacting with, for example, your lung cells, so it can't get in. And that's exactly how protection via antibody works, and how vaccines work. In fact, as we'll see in a minute, all vaccines currently in use against SARS-CoV-2, and in fact, almost all the vaccines in preparation focus on this protein, so-called the spike, which is the surface of the virus, and it's the one that the virus uses to get into the cell. So it makes sense to use that as the target. But there are two other branches of the immune system that one needs to be aware of. The first ones are what are called the innate immunity. So our body has a large number of, again, proteins, a little bit like hormones, which are released into the blood or into the tissue, which have a non-specific effect, some of them against viruses. And the most important of these is interferon. And actually, I mentioned already Paul Ehrlich as a German Jew working at the beginning of the 20th century. The interferons were discovered by two scientists whose name is virtually forgotten today, except among specialists, Alec Isaacs and Lindemann. And Alec Isaacs had also a very interesting story. His grandparents were Galician Jews who came to Scotland and had absolutely nothing. They lived in a tenement, had absolutely zero income. And then the son of the original people made money and sent his son to university. And he was one of the discoverers of the interferons. So, and very tragically, he died at 45, maybe one of the reasons why his name is little known. But so this is known as the innate immune system. It's non-specific. Any virus can be subdued by the interferons. But on their own, often they don't work. They sometimes need these other branches of immunity to work in parallel with them. And then the third one is what we call T cells, and it's the one that David Katz and I have really worked on for most of our career, and it's shown here. And I just want to show you, finish with by one little movie. So here, these big blobs here stuck down here, imagine these are cells infected with virus. So this might be, for example, your lung cells, which are infected with coronavirus. And then keep your eye on these little round things. These are white blood cells called T cells. And you'll see that they come moving along this arrow, and they'll come along and kill their target. They're called cytotoxic. They kill their target. And here they are, you see it, here it comes along, and then all of a sudden that cell just crinkles up and dies. This T cell injects it with something which kills its target. Well, we won't watch the rest of the movie, but the cells are gradually killed by these T cells. So that triumvirate, innate immunity, the non-specific immunity, T cells that kill infected cells. Once the cell has been infected in a virus, we have cells that can kill them. And of course, antibody together are what make up our immune system. So I'll hand over to David at that point. David, you're muted.
Thank you very much. Now, you've got a collection of building blocks together from Benny. And what I'm really going to add on to that is that the immune system is obviously an excellent defense mechanism against infection. But that immune system defense can itself have side effects, or that immune system itself can go wrong. And when you talk about immunopathology, you're really looking at those side effects. For example, if you start off with a virally infected cell and a lot of interferon is produced, that interferon may interfere with the neighboring cells, may cause effects on the neighboring cells. Those effects may be to recruit more good cells or good molecules that will act against the infection, but can also have manifestations that we would recognize as disease. I'm sure that many people in the audience know somebody who's had rheumatoid arthritis. And rheumatoid arthritis is often regarded as an example of where something, you don't necessarily know what it is, in the joint, it becomes the target of the molecules that are being produced as a result of an immune reaction. Sometimes this can be a bystander effect. Sometimes it can be a really major component. And again, taking the analogy of rheumatoid arthritis, we know now from research done by colleagues of Benny's and mine in past years, by Mark Feldman, we know that a molecule, a innate immune molecule called tumor necrosis factor, which got named because it was identified in tumor immunology, but which was really not, most important role was not in tumor immunology, was that if you put large amounts of it, it causes damage to the joints. And then instead of using a vaccination type protocol, you can use an antibody against the tumor necrosis factor, or an antibody against the receptors on the cells that bind tumor necrosis factor, and thus damp down and a bit of an immune response that you don't really like. So that's one way that one can understand immunopathology. The other way one can understand immunopathology is that when you get killing of large numbers of virally infected cells, as the movie that Benny showed illustrates, you get killing of these cells, but you also get release of virus. And if there a large amount of virus is released from the killed cells, then a virus becomes available for other cells to pick it up, and for therefore to exacerbate and extend the infection. Now, these may sound like nice details that people study, and they know that they exist, and that, you know, kind of people like me can look in laboratories and teach students about them to understand what's called the pathogenesis, how a disease develops. But we need to consider back to the process that we're actually talking about now. We're talking about the COVID-19 situation and to what happens when a virus called SARS-CoV-2 infects a person, and what responses there are to that, and how we can actually look at those responses and learn to actually improve the outcome of the disease. So on the one hand, we're going to have a vaccine strategy. The vaccine strategy helps you to understand how, if you get infected, you will be infected by a small number of viruses, and your memory of your antibody and your T cells will kick in, and they will remember that you've seen this before, they will produce the right molecules, and they will act very effectively. So that's the one way. But the other thing that might happen is that your immune system doesn't act as effectively, and under normal circumstances, when we haven't been vaccinated, that's what pertains. And the virus can win the battle. And winning the battle takes place, as we know from what we're seeing in the newspapers and the numbers and so on, quite often in the lung. And in the lung, you've got a whole complex of other individual cells, each of which will react slightly differently. But one of the important ways they will react is by producing a lot of these molecules, like tumor necrosis factor, like interferons, like other hormone-like substances like interleukins. And these themselves will cause damage. And in fact, the great evolution of our understanding of this aspect of immunology came many years after Isaac's. It's remarkable that he was able to identify and prophesy so far ahead. It came when the molecular revolution came into effect after the sequencing after the identification of DNA and RNA and making protein, when it became clear that each of these proteins would have a DNA sequence, an RNA sequence, and would produce a protein. And some people used to actually say at the beginning of that story, DNA makes RNA, RNA makes protein, protein makes money. And that's how the pharmaceutical industry came in. It's an interesting sideline at the moment, because of course we've gone one step further, because you might argue that some people have discovered that you don't have to go as far as the protein to start to make the money, because now for the first time, after many years, when people have been hypothesizing that it might be possible, RNA is actually being made into a useful form that might be important in forming vaccines. But the other element remains there, and it's quite interesting because when you look back to the dim distant past, which I'm talking about in February, and I had to talk about this a little. I asked two friends who I'd worked with a very long time ago, what they thought, if we weren't going to get a vaccine, what would we need to actually damp down some of these negative impacts? And all of us agreed that, there were steroids. Dexamethasone would be a good idea, but we were a little worried because sometimes steroids can damp down a good immune response as well as damping down an immune response that's gone out of control. Others would actually say, well, you should look at other different pathways. And one of my colleagues said, well, the drugs that are used in rheumatology, antibody against tumor necrosis factor, would be a good idea to look at them. And we see today that that's one of the drugs that are being tried and tried seriously, not like bleach. And a third colleague said, well, why is nobody looking at inhibition of one of the interleukins, which is the interleukin 6? And sure enough, today, 10 months later, anti-interleukin 6 antibodies are being seriously regarded as good news for use during the stages of the disease. So that's to supplement or to be an alternative to the vaccine strategy. But coming back to the question of the vaccine strategy and to what we're looking at, we are not only looking at chronic effects on the lung, at longer term effects on the lung. We've all heard of the term long COVID. We don't actually often define what we mean by long COVID. But if you look back at all the pandemics and viral infections there have been in the past, there very often have been long-term sequelae. People often forget that the consequence of the flu in 1918 were the famous patients that Oliver Sacks looked at. the patients with encephalitis lethargica. People often forget that the consequences of the polio pandemic were enormous. They were enormous in terms of disability and enormous in terms also of ethical dilemmas, because even in those days, there was competition for the intensive care units and for the intensive units, the availability of iron lungs, and indeed for the remarkable volunteering capacity of doctors and medical students when they actually used to be, if you couldn't get an iron lung, doctors and medical students used to act as human pumps to try and keep the lungs of people with polio going. So there are many, many traditions from the past of complications of viral infections actually lasting much longer than the immediate acute infectious phase. And that is what we are gradually learning, that long COVID may have lung manifestations, it may have kidney manifestations, it may also have probably cerebral manifestations as well. So those are important issues. Two final points. One is there is a cross communication, particularly in the lung and in the kidney, between the immune system and the coagulation system. And we know today that from the work particularly of the head of University College London Hospital, from Professor Levi, from his work, how this thrombotic tendency has meant that people have been even sicker than they might have been otherwise. So there are many, many areas of medicine which COVID-19 is impacting on, but the basic principle and the basic building blocks and the most basic tool that we can work with is to look at the normal immune system, to look how it reacts to infection, to look at how it may be modified by the disease, and then to see whether our vaccine strategies and treatment strategies combined can be implemented in order to alleviate and, if possible, treat this disease as well as to prevent.
Thank you very much for that. I believe, Professor Chain, that you're now going to present us again subsequent comments, but I can't see you actually on the screen. So, Professor Chain, are you?
Yes, I'm here. Sorry. Yeah, just very, very briefly, I just wanted to say something about the vaccines because I think it's one of the things that people are most interested in, so let me just. So I'm going to show you this, which is a very complicated slide. The only reason I'm showing it is that to emphasize that it's quite remarkable how in really only a year, well over nearly 100 different candidate vaccines have been tested or begun to be tested, have been developed against COVID-19. Of all sorts of different types. Perhaps some of you will know the story about Edward Jenner when he protected people against smallpox. He did it because very serendipitously, just by luck, there was a very related virus called cowpox, which was sufficiently similar so that the body got confused and made antibodies against the smallpox by giving the cowpox, which was a completely harmless virus. Now, we don't have the equivalent for SARS-CoV-2, or at least we don't know a related virus that is sufficiently close to use as a vaccine, but has no danger. But the approaches that have been used, there are three types of vaccine currently approved for use. The first one is the first one to have been released are the so-called RNA vaccines. David mentioned that this has been a real step change in vaccine technology. This has never been tried before for an infectious disease. The building blocks of the body are proteins, but they are coded for. The body produces those proteins according to a code, and the code is in the nucleic acid. So it's like a book that you can read the book and then, or instructions, and then make the whatever it is from the instructions. So similarly, the proteins are made from these instructions. Now, in the past, what we've done for vaccines is taken a protein and inject it and then get the body to make a response. With the RNA vaccines, we've taken the code for the protein, which is much, much faster to manufacture in principle, although there are many technical issues, and we inject that and get the body itself to produce the protein. In this case, the spike protein. All these vaccines are based on the outer protein of the of SARS-CoV-2, this thing that's sticking out here, shown over here. So that's the Pfizer vaccine and the Moderna vaccine are both examples of these new, completely new type of vaccines, which may well revolutionize vaccine technology in the future. I say we don't have a virus like Jenner had cowpox. We don't have a convenient similar virus to trick the immune system with. But what we can do, because of molecular biology, the revolution in molecular biology over the last 50 years, is we can take a totally harmless virus, in this case, for example, a virus called an adenovirus, a monkey adenovirus, which infects humans but causes no disease as far as we know. And we can insert into that virus a little bit of SARS-CoV-2, in this case, the code for the spike protein again. So we've sort of done what Jenner did, but in a rather fancy way. We've taken a harmless virus and inserted a piece of SARS-CoV-2 in it. And that is, of course, the Oxford/AstraZeneca vaccine, and also the one-shot Johnson & Johnson vaccine, which will, has just released its phase 3 results. And then the final vaccine, the latest one to be released, is actually the most traditional type of vaccine. Here we've done exactly what has been done with tetanus, with polio, with diphtheria. We basically take the building block, the spike protein, this knob on the surface of the virus, and we make it in a large test tube, a series of test tubes. We call them bioreactors because they're so big, but I mean, in essence, it's a kind of a very big test tube. And we make them synthetically, and then we inject that protein so that the body thinks it's being attacked by the virus, but in fact, it's being not really attacked, but it's being exposed to this harmless little piece of virus, which looks like the real thing, and the body responds to it. So those are the 3 types of vaccines which are now approved and of course are being rolled out with extraordinary efficiency, actually, in the UK and also in Israel. So I'll stop there. And yeah.
Thank you very much. Thank you so much, both Professor Katz and Professor Jain, for your presentations. I've had a flurry of texts via the chat of questions, but please do, to everyone really, please do feel free to share more, and I will try and pass them on. I'm not going to direct the questions specifically to either of you because I'm not sure I know enough the different areas of expertise to do so, but I'll ask the questions and perhaps you can yourselves decide whether to who is going to answer. The first question is about age sensitivity and the immune system. Is it that the immune system is always so age sensitive? Why is it particularly with COVID that this seems to create such a different effect? Is it about the reaction to COVID or the effectiveness of the immune system? That's the first question that I was asked via the chat group.
David, do you want to answer that?
Well, yes, and am I muted or not?
No,
yeah,
no, we can hear you.
We can hear you.
It's an interesting question. First of all, of course, the age does affect the immune system. You know, your immune system efficiency and so on naturally wanes. That's number one. But number two, we mustn't confuse age with what are called comorbidities. there are lots of other things that may account for that age effect, other than actual chronological age. We know that older people were more likely to actually be potentially dying of the disease, but they may be that they had less spare respiratory reserve function, and they had less mobility of their lymphocytes or other cells than younger people. That doesn't mean that age is actually the factor. And kind of the diabetes, high blood pressure, overweight, and the social inequalities, as Sir Michael Marmot would say, are the factors that really make a big difference, and not necessarily just age per se. And he produces graphs which show what I've just said, which show that if you look at populations and you correct for age, you find that it's the inequalities between the populations and not the age that is the determining factor.
Thank you for that. I've had a flurry of questions around both the theory and the practice of the booster, the second dose. First of all, what's the mechanism that the first dose doesn't bring complete cover and then the second one does? And secondly, what is your view about the 12-week delay as opposed to the 3-week delay between the two doses? And thirdly, I guess on a practical level, your view around mixing and matching. So if one has a first shot with one of the vaccines and then a second shot with another one, for example, what's again, theoretically and practically, what the differences are and how that works?
I'll take that one, David.
Yeah, sure.
So first of all, the theory of boosters. If we go back to what I was saying in the video, in order to make an immune response, you have to find that antibody, which is an exact match to whatever the challenge is, in this case, COVID. Now, you obviously have to, in order that you've got one antibody at least for every possible pathogen that you might ever encounter, any virus, any bacteria. You have millions of different ones, but the quid pro quo for that, the sort of downside, is that you only have very little of each one to start with, because there wouldn't be room for everything. After you've been exposed to either an infection or a vaccine, you then make many more of the cells which were specific for that particular thing that you were exposed to the first time. So that it's called a secondary immune response, so that when you re-expose yourself, the amount of antibody that you make is much larger than in the first round. And it's also qualitatively better. It fits better. It's a better fit to the antigen. I mean, the data on, for example, the Pfizer vaccine suggests that you make at least 100 times higher vaccine, higher level of antibody after a second response. Now, what we don't really know is what is enough. You know, how much antibody in the blood is enough to protect you. But the idea to having boosters, and most vaccines, not all, but most vaccines are multi-shot. I mean, it's true about most of the childhood vaccines. And the reason is because, as I say, the second response is always much stronger than the first response. In terms of mixing and matching, I doubt whether that will be done in practice until it's actually been tested, because it's an untested thing. But in principle, I would imagine that they would work extremely well. In some cases, they may even work better for various technical reasons, to give, for example, the Oxford vaccine first and the Pfizer vaccine as a booster might in certain conditions be actually better than giving 2 lots of the Oxford vaccine. So that was the second. Was there a third thing, Rabbi Zobin, you said?
I think there was just a question around the government policy of 12-week as opposed to a 3-week delay.
So this is a very, very difficult and quite controversial point. At the moment, the UK is the only country in the world to have taken that decision. Basically, what the government have to, what the committee, in this case, it was a joint vaccine Joint Committee on Vaccination, that is a regular standing body that advises the government on vaccination, they took this decision. What they had to balance was as follows. The danger is that a new variant of the virus will emerge, which is more virulent, but more worryingly might be resistant to the established immunity. So it could reinfect people that have been already infected, or it could be resistant to the vaccines. Now, the longer you leave it and the more chance the virus has to divide and go from person to person, the more options it has for such a mutation to turn up. And once it turns up, it will, of course, be selected because it will be an advantage versus the original virus. So you need to stop the virus circulating as quickly as possible. But what the government had to balance was, is it better, do you prevent the virus more by giving more people one dose so that the more people will be at least partially protected, and therefore you'll cut down the number of transmissions in that way, or to immunize a smaller number of people, but more effectively, so you really close down the virus. The danger with one shot is that during those 12 weeks, it's not that the second dose won't work after 12 weeks, it'll work, if anything, better, certainly not worse than after 3 weeks, but it's a 12-week window during which, if your immunity is suboptimal, there's a genuine danger that the virus, that a mutated virus, and that could, of course, cause havoc with the whole system. It was a really difficult call. I don't have enough data to say, you know, which one was, whether it was the right call or the wrong call. I don't think anybody does at the moment, actually. But that was the balance they had to come up with.
Thank you. There have been a number of follow-up questions actually relating to your answer as you were speaking, including whether antibodies from being infected, is that equivalent to the vaccine? And would if one has antibodies from being infected, would that effectively match the first vaccination such that the vaccination one gets is equivalent to the second vaccination? In brief, how the two interact?
That's an interesting question. We don't know really, but I would say yes, probably. I mean, in other words, if you've been vaccinated, you will have in most cases, about over 90% of cases, you will have a good antibody response. And the data suggests that very few people are being reinfected and getting sick. It's a really very low probability. If you've been, if you have had the response, if you've had the virus and then you get the vaccine, it's quite likely the vaccine will act as a sort of as a booster essentially, and you'll just boost your antibody response again. There's nothing to suggest that the antibody response to the vaccine is substantially different to the antibody response to the virus. Except in the case of the real virus infection, you actually get antibodies to all portions of the virus, not just the outer spike protein. As whether those have any protective value at all, or they're just a side effect, or they could even be, as David explained, a sort of bad side effect, we just don't know at all.
And again, I guess somewhat of a follow-up to that is how long will immunity last for? Do you anticipate this being annual? And why is it immunity wears off? Is it that the body forgets its lessons that are learned, or something else at work here?
Oh, David, you try answering that question.
Well,
it's a hard one.
While I was listening to the questions, there's one thing that you hear the questions and you realize things that you should have possibly said. One of the things that relates to this is our knowledge about vaccines from the past. You know, the polio vaccine. Remember, we get vaccinated against polio and we don't have it, it doesn't happen again. Hepatitis B, a different virus, a different kind of vaccine, but also a vaccine that we get 3 shots of. papillomavirus, yet a different kind of vaccine. We're quite used to, in the system, having different vaccines for different kinds of virus, let alone different kinds of organisms. I'm not even starting on meningococci and things like that. So it's not a surprise to me. It would be a surprise to me if there wasn't uncertainty about whether you'd have to have annual immunizations or not. The annual one that we all know about is flu. And there, the reason is that the flu virus changes and is not the same. And there are two particular proteins, the H protein and the N protein, they change every year. Now, we don't know we know an enormous amount. Just look at the list, look at the known sequences that we can identify these mutants. Is itself an extraordinary achievement in this space of time. But we don't know yet whether those mutants are actually going to follow through, and something like that change is going to happen in every year, and it's going to be insufficiently critical a part of the coronavirus that it will need introduction of a new vaccine. What I will say is that, again, the reason why the RNA technique is so remarkable is because it's not difficult to work out how we could do that. We could produce a slightly different RNA vaccine, sufficiently different that it will prime a different set of antibodies and be successful. So that's the difference. Is we have the tools to ask that question in a much more scientific way than we've been able to do in the past. I think is that a reasonable answer for the moment?
Yeah. Yes,
Thank
it's
you.
a difficult question.
Thank you for that. I'm conscious of the passage of time, so I'm going to prioritize two practical questions, and then if there's time, we'll ask a couple. There's a variety of theoretical questions that are coming through. The first practical one, I think that is a very important question for us to hear your views on. Do you have any concerns about the speed of development of the vaccine? 10 months versus perhaps 10 years normally. Can we be sure about long-term effects of the vaccine? How can scientists reassure us about that? And I think that's probably an important question for us to hear your thoughts about that.
I'll have a go at that. It has been an incredibly fast production schedule from sort of conception to actually rolling out. But I think the thing to bear in mind is that although this specific vaccine target is new, what this builds on is really, a century of well-established technology in the development of vaccines of different types, as David said. So I see no reason to think that there should be any particular danger to any of the types of vaccines that are being rolled out now compared to the standard vaccines. I said that RNA vaccines are a completely new introduction. That's true in terms of large-scale rollout, but the science of RNA vaccines has been around 20 years or more. It's being tried in the context of cancer, which is a very hard nut to crack from the vaccination point of view, the idea that you could vaccinate against cancer. And that's nothing to do with the fact that it was an RNA vaccine, just the principle of vaccination is very hard in that context. But there's really a wealth of experience of RNA vaccines in various trials over those 20 years. So I personally think it's entirely safe. I don't think there's any, one can never say with 100% certainty anything, but I can see no obvious reason why any of the vaccines being trialed are in any way dangerous. The Oxford vaccine has actually been tested in a modified form for Ebola. The Ebola vaccine was a very similar vaccine and was given to quite a large number of people actually at the time of the Ebola outbreak a few years ago. And there's been no report of side effects other than the normal side effects of vaccines, which is, sometimes you get pain at the site of infection and occasionally slight fever following particularly the booster. Actually, rumor has it that there is quite a strong sort of kind of achy type flu-y feeling for boosting of the Pfizer vaccine, perhaps because it's such an effective vaccine, it gives a very strong response. But I don't think there's any really reason to be wary of this vaccine, just as there isn't in general for vaccines. They're actually, if you look at the safety record of vaccines, I mean, they are among the safest medical interventions that has ever been introduced. Literally millions and millions and millions of people have been taken various vaccines, and the number of side effects is really small.
Thank you for that, and I guess really important for all of us to hear that reassurance that this is a very, very well tested form of medical intervention. Thank you. Again, just two very practical questions. The first is, Israel is now encouraging pregnant and breastfeeding women to be vaccinated. Britain isn't. Why is that, and what would be your personal advice around that?
David, do you want to say something on that?
Well, I mean, I've never understood exactly what the reason for the not vaccinating pregnant women at all. I find it very difficult. There is some story about a computer identifying shared sequences between viruses and some proteins, but I've never understood that. And as far as I'm aware, there is no data that it is unsafe in pregnant women. I think the Israeli suggestion is correct.
Thank you. And finally, I guess in terms of practical questions, I'm very conscious of the passage of time. And I do apologize to the people who have been asking questions in the chat for whom we're not going to get time to answer. The question here, is there anything we can do to boost the effectiveness of our immune system? Vitamin D, effective, not effective, just any practical advice you can give us in general for our health of our immune system?
Well, I mean, I think in general a good diet is good. It's good for everything more or less, and the immune system is no different. There is a specific story around vitamin D and COVID-19. There have been stories around vitamin D and various other infections, and certainly I think it plays a role. Whether taking supplemental vitamins will actually help, I don't think the data is really there. I don't know if David knows of any other data.
No, I agree with you on that. There's no specific data there.
No, and I don't think anything else I can't really think of other than, living a healthy life is a good idea from every point of view. I mean, the immune system works best. The immune system is one of the most expensive systems of the body to run. It equals the central nervous system, that's to say our brain. The body devotes an enormous amount of its energy on a day-to-day basis. Those white blood cells that I was telling you about, they get replaced on a daily basis in millions and millions as they get broken and are replaced. So anything so that means that we are devoting a lot of our energy, just even if we're not doing anything, to running an efficient immune system. And that means that the less that we divert to other things to try and combat other things going wrong in our body, the better the immune system will function. It's a kind of a trade-off. If you're so busy dealing with being overweight or diabetes and so on, then the immune system inevitably suffers.
Thank you so much for those answers, and really thank you for this evening. I once again apologize. The number of questions and comments that have come through is simply staggering. And I'm sorry to those who have asked them that we're not able to address them all, but I'm also being flooded with, in the chat with expressions of thanks and appreciation. I think this was so interesting for us to hear and to understand a little bit better about what's going on in this world. On a practical level, the take-home message also was light now and clear from two really eminent experts in the field who we trust absolutely, who are reassuring us and telling us this is absolutely the right step to take. The figure, perhaps Professor Chain, that really struck me was this figure of 300 million smallpox deaths, and us realizing the power of this technique to save lives is remarkable. And thank you so much for educating us and informing us about this area and the that God has given human beings the wisdom and insight to achieve such understanding. I want to conclude by thanking again our eminent guests, thanking Julie Conn for both organising this evening and for everything else that she does for education in the community. A quick plug, Wednesday evening this week, we have the KLBD, the London-based virtual roadshow, to help us understand the world of kashrut, and a date for the diary, Sunday, the 14th of February. We have an international Yom Limud pre-Purim, so many things happening. Please look at our shul calendar online and the emails to see so much that's going on over the next few weeks, but these are just two things that I'll very quickly plug. And thank you so much again to our visitors, to our guests, Professor Katz, Professor Chain, thank you for the time and energy and insight you gave to us. And thank you so much to everyone for joining this evening.
Thank you. Thank you very much.